Section 17 of Gray's Anatomy Part 4. This is LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Fore-brain Part 5. Structure of the cerebral hemispheres. The cerebral hemispheres are composed of gray and white substance. The former covers their surface and is termed a cortex. The latter occupies the interior of the hemispheres. The white substance consists of medullated fibers varying in size and arranged in bundles separated by neuroglia. They may be divided according to their course and connections into three distinct systems. 1. Projection fibers connect the hemispheres with the lower parts of the brain and with the medulla spinalis. 2. Transverse or commissural fibers unite the two hemispheres. 3. Association fibers connect different structures in the same hemisphere. These are, in many instances, collateral branches of the projection fibers, but others are the axons of independent cells. 1. The projection fibers consist of efferent and afferent fibers uniting the cortex with the lower part of the brain and with the medulla spinalis. The principal efferent strands are 1. The motor tract, occupying the genu and the anterior thirds of the occipital part of the internal capsule, and consisting of A. The geniculate fibers, which decussate and end in the motor nuclei of the cranial nerves of the opposite side, and B. The cerebrospinal fibers, which are prolonged through the pyramid of the medulla oblongata into the medulla spinalis. 2. The corticopontine fibers, ending in the nuclei pontis. The chief afferent fibers are 1. Those of the lemniscus, which are not interrupted in the thalamus. 2. Those of the superior cerebellar peduncle, which are not interrupted in the red nucleus and thalamus. 3. Numerous fibers arising within the thalamus and passing through its stalks to the different parts of the cortex. 4. Optic and acoustic fibers, the former passing to the occipital, the latter to the temporal lobe. 2. The transverse or commissural fibers connect the two hemispheres. They include A. The transverse fibers of the corpus callosum, B. The anterior commissure, C. The posterior commissure, and D. The lyra or hippocampal commissure. They have already been described. 3. The association fibers unite different parts of the same hemisphere and are of two kinds. Those connecting adjacent gyri, short association fibers. 2. Those passing between more distant parts, long association fibers. The short association fibers lie immediately beneath the gray substance of the cortex of the hemispheres and connect together adjacent gyri. The long association fibers include the following. A. The uncinate fasciculus. B. The cingulum. C. The superior longitudinal fasciculus. D. The inferior longitudinal fasciculus. E. The perpendicular fasciculus. F. The occipitofrontal fasciculus. And G. The fornix. A. The uncinate fasciculus passes across the bottom of the lateral fissure and unites the gyri of the frontal lobe with the anterior end of the temporal lobe. B. The cingulum is a band of white matter contained within the cingulate gyrus. Beginning in front at the anterior perforated substance, it passes forward and upward parallel with the rostrum, winds around the genu, runs backward above the corpus callosum, turns around the splenium, and ends in the hippocampal gyrus. C. The superior longitudinal fasciculus passes backwards from the front lobe above the lentiform nucleus and insula. Some of its fibers end in the occipital lobe and others curve downward and forward into the temporal lobe. D. The inferior longitudinal fasciculus connects the temporal and occipital lobes, running along the lateral walls of the inferior and posterior cornues of the lateral ventricle. E. The perpendicular fasciculus runs vertically through the front part of the occipital lobe and connects the inferior parietal lobule with the fusiform gyrus. F. The occipital-frontal fasciculus passes backward from the frontal lobe along the lateral border of the caudate nucleus and on the mesial aspect of the corona radiata. Its fibers radiate in a fan-like manner and pass into the occipital and temporal lobes lateral to the posterior and inferior cornua. Desjardins regards the fibers of the tapetum as being derived from this fasciculus and not from the corpus callosum. G. The fornix connects the hippocampal gyrus with the corpus mammillare and, by means of the thalamo-mammillary fasciculus, with the thalamus. Through the fibers of the hippocampal commissure, it probably also unites the opposite hippocampal gyri. The gray substance of the hemisphere is divided into 1. That of the cerebral cortex, and 2. that of the caudate nucleus, the lentiform nucleus, the claustrum, and the nucleus amygdalae. Structure of the cerebral cortex. The cerebral cortex differs in thickness and structure in different parts of the hemisphere. It is thinner in the occipital region than in the anterior and posterior central gyri, and is also much thinner at the bottom of the sulci than on the top of the gyri. Again, the minute structure of the anterior central differs from that of the posterior central gyrus, and areas possessing a special type of cortex can be mapped out in the occipital lobe. On examining a section of the cortex with a lens, it is seen to consist of alternating white and gray layers thus disposed from the surface inward: 1. A thin layer of white substance, 2. A layer of gray substance, 3. A second white layer, outer band of Ballinger or band of Generi, 4. A second gray layer, 5. A third white layer, inner band of Ballinger, 6. A third gray layer which rests on the medullary substance of the gyrus. The cortex is made up of nerve cells of varying size and shape, and of nerve fibers which are either medullated or naked axis cylinders embedded in a matrix of neuroglia. Nerve cells. According to Cajal, the nerve cells are arranged in 4 layers, named from the surface inward as follows: 1. The molecular layer, 2. The layer of small pyramidal cells, 3. The layer of large pyramidal cells, 4. The layer of polymorphous cells. The molecular layer. In this layer, the cells are polygonal, triangular, or fusiform in shape. Each polygonal cell gives off some 4 or 5 dendrites, while its axon may arise directly from the cell or from one of its dendrites. Each triangular cell gives off 2 or 3 dendrites, from one of which the axon arises. The fusiform cells are placed with the long axis parallel to the surface and are mostly bipolar, each pole being prolonged into a dendrite which runs horizontal for some distance and furnishes ascending branches. Their axons, 2 or 3 in number, arise from the dendrites and, like them, take a horizontal course, giving off numerous ascending collaterals. The distribution of the axons and dendrites of all 3 sets of cells is limited to the molecular layer. The layer of small and the layer of large pyramidal cells. The cells in these 2 layers may be studied together, since with the exception of the differentiation in size and the more superficial position of the smaller cells, they resemble each other. The average length in the small cells is from 10 to 15 microns, that of the large cells from 20 to 30 microns. The body of each cell is a pyramidal in shape, its base being directed to the deeper parts and its apex towards the surface. It contains granular pigment and stains deeply with ordinary reagents. The nucleus is of large size and round or oval in shape. The base of the cell gives off the axis cylinder, and this runs into the central white substance, giving off collaterals in its course, and is distributed as a projection, commissural, or association fiber. The apical and basal parts of the cell give off dendrites. The apical dendrite is directed towards the surface and ends in the molecular layer by dividing into numerous branches, all of which may be seen, when prepared by the silver or methylene blue method, to be studied with projection bristle-like processes. The largest pyramidal cells are found in the upper part of the anterior central gyrus and in the paracentral lobule. They are often arranged in groups or nests from 3 to 5 and are named the giant cells of Betz. In the former situation, they may exceed 50 microns in length and 40 microns in breadth, while in the paracentral lobule, they may attain a length of 65 microns. Layer of polymorphous cells. The cells of this layer, as their name implies, are very irregular in contour. They may be fusiform, oval, triangular, or star-shaped. Their dendrites are directed outward, but do not reach so far as the molecular layer. Their axons pass into the subjacent white matter. There are 2 other kinds of cells in the cerebral cortex. They are A. The cells of Golgi, the axons of which divide immediately after their origins into large number of branches, which are directed towards the surface of the cortex. B. The cells of Martinati, which are chiefly found in the polymorphous layer. Their dendrites are short and may have an ascending or descending course, while their axons pass out into the molecular layer and form an extensive horizontal arborization. Nerve fibers. These fill up a large part of the intervals between the cells and may be medullated or non-medullated, the latter comprising the axons of the smallest pyramidal cells and the cells of Golgi. In their direction, the fibers may be either tangential or radial. The tangential fibers run parallel to the surface of the hemispheres, intersecting the radial fibers at right angles. They constitute several striata